And Adrian and I had the opportunity to meet some of you in Bariloche last November during the Palynology conference, which was very nice. But we missed many other that couldn't attend, so we are very happy to restart this online activity and see more people joining us. Our speaker today is Dr. Paul Zander, and I'm so pleased that Paul accepted to give this talk because I have been following his research in the last years and he's doing very innovative and promising stuff. So a little bit about Paul. He completed his degree and master at Northern Arizona University, where he did his dissertation on tephrochronology and paleoenvironmental reconstruction from lakes in Alaska. And after that, he worked as a research assistant at the University of Arizona and then moved to Switzerland to start a PhD on barf of Lake Sabinsky, supervised by Martin Grossian and Wojciech Tillman. So currently, Paul is a postdoc at the Max Planck Institute for Chemistry in Germany. And during his career, Paul has specialized in geochronology, organic geochemistry, high-resolution core scanning techniques. And today he will tell us about his research during his PhD on hyperspectral imaging for the biochemical analysis. So Paul, it's all yours.
Great. Thank you so much for that nice introduction. Thanks for inviting me, and thanks everyone for coming. Can you all see my screen and the laser still? I think it's still working. So today I'm going to talk about hyperspectral imaging, sediment core scanning for biogeochemical analysis. And this work was basically done at the University of Bern as part of my PhD, and while I was there as a postdoc, and also in collaboration with other people from University of Gdansk and ETH and so on. Now I'm at Max Planck Institute. I'm not really actively working on hyperspectral imaging here, but I'm still involved in projects using the hyperspectral scanner that they have in Bern. And this is an image of the hyperspectral scanner in Bern that we've been using. So let's get into it. There's going to be kind of two parts to the talk. In the first part, I want to give a background, tell about the origin of the method, go into the details of how the method actually works in terms of the workflow, talk about the uses and the strengths and limitations of this hyperspectral imaging method. And then in part two, we'll go into example applications, show what's been done on lake sediments, and I'll use some examples from other people's work, but then have an emphasis on some of my most recent work as well. In particular, a recent study where we show a new type of pigment that can be detected with this method from green sulfur bacteria. Okay, so background. Where did this method come from? There was kind of a previous method called visible near infrared spectroscopy. And the idea here to understand is we are measuring light reflected off of a sediment core. So you measure light across a spectrum of wavelengths, and you see that this can tell you something about the composition or properties of the sediment depending on which wavelengths absorb more or less light. And this was initially done with point measurements with tools such as the Spectrolino or this Minolta camera, which is often mounted to the multi-sensor core loggers. So people have been obtaining this data on sediment reflectance for a while. And one of the first studies to show how useful this data could be was from Ryan and Sirocco. They showed that this absorption trough here, so we're looking at wavelengths of light, and more light is absorbed in this area around 660 nanometers. That's because there are chloropigments, chlorophyll in the sediment that absorbs this light, making the green color. And they could show in this study that more absorption, a deeper trough meant there was more chloropigments, and that could be used to track past productivity, right? But these technologies were somewhat limited in terms of the resolution. So the measurement field was 2 to 8 millimeters, still pretty high resolution data when you're talking about geochemical data from lake sediments. The spectral resolution was only about 10 nanometers across this spectrum of light, and the spectral range was 360 to 740 nanometers. So that's sort of the old version. And then already with that technique, people were finding really interesting results, such as you could relate this RABD 660, 670, which is essentially measuring how deep that trough is, I showed on the last slide. You could relate that to temperature in some cases. This is an example from a lake from Chile, where the RABD is plotted in gray and summer temperatures are plotted in black, and you see a pretty close agreement between the two. So this showed potential for this method, in particular because it provided such high resolution data for making time series, which is really important when you want to do calibrations with instrumental climate data. So the next advance was to use hyperspectral imaging. Similar concept, you're measuring light reflected from a sediment core, but it's more advanced because now we are taking an image. So we have a two-dimensional area that we can measure data from. And the main advance is that we have much higher resolution data using this technique. So here's again an image of the core scanner we're using in Bern. We have a prototype of this core scanner that's made by the company Specim, and now they are making this commercially available. And what happens is you put a sediment core in the sample tray passes underneath this whole apparatus, which contains an illumination unit here in orange, shining light down onto the sediment core, and the hyperspectral camera up here. And you can get different types of cameras that have different types of sensors measuring different areas of the light spectrum, right? So there's the visible near infrared sensor, which would be this one, covering a spectral range of 400 to 1,000 nanometers, or you can have shortwave infrared going further into the infrared, longer wavelengths, 1,000 to 2,500 nanometers. The one that I have worked directly with is the visible near infrared one, so that's where I'll spend the most time, but they both can be useful. And the way the image is acquired is these sensors are really row line scan sensors. So the core passes underneath the camera, and as it passes through, the camera acquires single lines of pixels that are then stacked on top of each other. This is called the push broom style image acquisition to create a complete picture of the core. And you can control different parameters. Changing the camera height changes the field of view, so how much of the core you see, and it changes the spatial resolution of the image as well. So a higher camera height means you have a lower resolution image. All of these parameters need to be kind of aligned, need to be compatible. So the field of view, the camera height, the scanning speed, how fast the core goes through, and the camera exposure all need to be in agreement so that you get an image that's not distorted. And the big thing with the camera exposure is it depends on how bright or dark the sediments are. You might need to have a longer exposure time with very dark sediments, and that means slower scanning speed, right? But in general, scanning a 1 meter core takes about 30 minutes and produces a 20 gigabyte raw image file. Yes, so that image file we call a hyperspectral data cube because it has these 3 dimensions. It has the 2 spatial dimensions, X and Y, on the core face. And then for each X, Y position, there's also a Z dimension, which is the reflectance across the light spectrum, right? So if you go to each XY coordinate, you have a pixel, and within that pixel, you have a profile that looks something like this, right? Also, I wanted to show it here. This would be an example profile from a single pixel where you can see how the light absorption changes across the spectrum at a particular pixel. So we get this all across the core image. It looks like this. And so let's go through the workflow, how the method works. First step is data acquisition. That means preparing your sample, cleaning the surface of the sediment core, making a smooth, clean surface, determining the camera and scanner settings. As I said, it depends mainly on the brightness or darkness of the sediment. Then you take your image and Next, you go into the data pre-processing phase. The first step is always normalization. So at first you get this raw data file of reflectance, and we compare that reflectance to standards, dark and white references. The dark reference would be the camera with the shutter closed, so no light entering the camera whatsoever. And the white reference is a barium sulfate plate that we take an image of that's very white, and we consider that to be 100% reflectance. And then all of our data gets kind of scaled to these two endpoints, and it can be interpreted as a percentage between that dark reference and the white reference. So that's the normalization step. Then we define regions of interest. There's different ways to do this, but typically we define a big area that covers the nice part of the core, leaving out the edges. And this is where we will calculate things on in our next step. And then we often also define a smaller, narrower region of interest, usually like a 2 millimeter wide rectangle going down the core. It can also be 5 millimeters wide, that will capture kind of the nicest part of the core, and where we can do more computationally intense things on this smaller region of interest. And then we also use that usually to extract our down core time series so that they come from one specific area that's nice and clean, and that way we don't have any problems with varves at an angle or bended varves or anything like that. Then you can do other pre-processing steps like transformations, like continuum removal or taking derivatives of the spectra. You can do filtering or destriping, smoothing of the spectra. These are all different pre-processing steps you can do depending on the quality of your data or what you're looking for. And then you go into data post-processing, so really learning something from the data. And a first step we typically do is to extract spectral end members. So these would represent sort of the pixels in your image that are most different from one another in terms of their spectra. These should represent relatively pure areas of the sediment, so where you have like the most clay or the most calcite or something like this compared to the rest of your core. And this just gives us an idea of the variability within a core, and it starts to help us to see what kinds of features are there in the spectral data that we might want to quantify, right? So changes in slope or troughs, things like this, right? So it starts to give us an idea of what's in the sample. Based on that, you can select spectral indices to calculate. So we often calculate these relative absorption band depths, measuring how deep a trough is. You can also measure trough areas. You can, there's indices that are simple ratios between 2 points on the spectral profile. And if you want, you can combine that with measured properties of the sediment and do regressions or calibrations. Calibrations can also be done without calculating an index, but simply from the full spectral data. Or another kind of way of processing this data is through classification, which means sort of grouping pixels into different classes. This can either be based off of the spectral end members, or it can be based on defined areas within the core. So you can go to a certain layer and say, this layer looks like a flood layer, and use an algorithm to classify all pixels that are similar to that layer throughout the rest of the core, for example. In any case, no matter what method you use, you can obtain both spatial maps of whatever parameter you're looking at and down core time series data, right? So here's some examples of what that can look like. You have the spatial data and you have this down core time series data. Right, in our group in Bern, one of the things that we have been working on to get a better understanding of is doing quantitative calibrations, comparing typically relative absorption band depth indices to pigment concentration. So I want to walk you through how we are typically doing that. So we measure these, we quantify these troughs using an index where we would draw a line that would represent perhaps what the spectra would look like if there was no pigment there. And then the RABD index basically measures essentially it measures the depth here between the continuum of what it would have been like without a pigment to what it is now with that absorption feature, right? So this RABD 670 corresponds to the chlorophyll A, and the RABD 845 corresponds to bacteria phaeophytin A. I'll talk about both of them later. So we measure that throughout the core, and then we'll take samples from specific places along the core, usually trying to cover a full range of very low RABD values and very high RABD values. We take samples, typically 1 centimeter square or 1 centimeter cube samples, and extract them with solvents and measure the pigments using either an HPLC or a spectrophotometer so that we can get the actual concentrations. And then you can do a calibration using linear regression. Here's an example with the RABD 845 and bacteria phaeophytin A concentrations. And you see that there's a linear relationship between the two. And we're typically getting errors in the range of 10, maybe 15% with just looking at the calibration uncertainty, right? And with that, you can then translate your RABD data that you took from the whole core, pixel by pixel. You can translate that into a concentration. Here, total chlorophyll A, here, bacteria phaeophytin A. So now we have these spatial maps and these time series of pigments that are estimated in concentration. So kind of semi-quantitative concentration data at extremely high resolution. What I should mention that I think I maybe didn't say explicitly before, it was on a table back there, is the resolution of the image itself. How high resolution image can we get? We are taking images where each pixel is something between 50 micrometers to 100 micrometers. So that's 0.05 to 0.1 millimeters. So that is the resolution of the data that we can obtain at that scale, sub-millimeter scale. Okay, so what has been done so far with this method? I put together a review specifically about hyperspectral imaging on lake sediments, and this is the result that we got. You can see it's a new method that's growing, and people have been using it for both organic and inorganic proxies. The pigments are very popular, especially chloropigments, because they are present in so many sediments and they are easy to measure with this technique using the absorption troughs. People have also shown, or it looks like you can measure phycocyanin. This is a different kind of pigment that comes from cyanobacteria. This has been shown in a lake from Tibet, and we have shown also with some standards in our lab that this is working, although a true calibration has not been published yet. People have also measured aromatic organic matter, terrestrial organic matter. This can be measured in the shortwave infrared range, I believe. And people have made calibration models, regression models using the full spectral range, not a specific absorption band, to estimate bulk organic matter in sediments. People have also interpreted hyperspectral data for proxy of charcoal. And then in terms of inorganic proxies, you can detect lithics or clay minerals, calcite, just based off of total brightness, actually, total reflectance. There are calibration models to estimate grain size distributions. You can distinguish flood deposits using classification techniques, and tephra have also been distinguished. So you can see it's a wide variety of proxies that have been used with this method. So what are the strengths of the method? The big strength to me is that it's relatively fast and easy, and it spans this micrometer to meter scale, right? It gives you the detail at micrometer to do really detailed investigations of varves. Like you can see in this image here, I have 4 varves from Lake Shabinsky, comparing pigment data from hyperspectral imaging data with micro XRF from the Bruker Tornado XRF imaging system, which I think is a great combination because you get this high resolution spatial data from both. So you can look at these seasonal scale sedimentation processes, but at the same time, you can also look at a record that's 20 meters long, covering the full Holocene, and look at trends in productivity and pigments over these really long timescales, right? And it's it's a rapid enough method that you can actually apply it to records that are 20, 30, 50 meters long, and it's feasible. So that's the big advantage to me, is bridging those scales of micrometers to meters. A big strength is you can scale up the resolution of your conventionally measured data. So you can measure organic matter or grain size or pigments at low resolution throughout your core, develop a calibration model, and produce high resolution data throughout the core. And the combination with Varves is really powerful. As you see in this example, you can look at the seasonal scale sedimentation. This can tell us something about perhaps the influence of seasonal weather or aquatic chemistry and these cycles within a single year, right? And the spatial data is also valuable, right? It can tell you something about sedimentation processes or data quality compared to these line scan techniques where you just get, you know, a single profile. If you have an outlying data point, you don't know how representative that is. But if you have spatial data and you see a full layer, it gives you much more confidence in that data. And you can see the difference between a concretion versus a layer, right? And it gives both simultaneously organic and inorganic proxies, which can be very useful. As I said, I think the combination with other techniques like XRF, mass spectrometry imaging is a new technique. These have the potential to give us really a new level of detail when we're studying our Varves with quantitative data. But there are also limitations. There are matrix effects, so meaning other sediment properties that you might not be interested in can modify the spectral signal. Water content can have an impact, and just anything else in the sediment can change how a certain feature looks. Extremely dark sediments are a problem for this technique because we just don't have enough light reflected off of the sediments. We get really noisy data. And there's a problem with this technique, which is you can have non-unique spectral features, meaning a certain absorption area or a change in slope can be attributed to multiple different reasons, and you don't necessarily know, just looking at the spectral data, what's causing that feature in your core. So this is why it's important usually to have independent validation where you measure whatever property you're interested in with another technique to validate the hyperspectral data. And that also means we typically need site-specific calibrations to have precise results. I think it might be possible to have some generalized calibration models for something like chloropigments, which are present in a lot of places, but you're generally going to get a much more precise calibration model if you do it on each site specifically. Okay, so that was part one. And now I want to move into part two, which is looking at examples of applications. Not all of these are from my own work. Some of these are from other people's work. Like this example on recognizing tephra in Antarctic lakes. In this study, they went to some sediment cores that have clearly visible layers of tephra, like you can see these here. And they took hyperspectral images of the core and used a classification algorithm where they selected obvious tephra layers, known tephra layers, and said search for, in the rest of the core, other pixels that have a similar spectral profile to the spectral profile within this tephra layer. And the results come out in these images here. It's basically showing the similarity between the target and the unknown pixels. And so you see that it detects the obvious tephra layers very well. In fact, it was able to detect all of the tephra layers that were recorded by visual inspection. And it also highlights some areas that were not detected by visual inspection, like this sort of fine layer here, which might be kind of a diffuse tephra. So it can help to recognize tephra layers that might not be seen just by a visual core inspection. It's not going to be at the level of doing a full cryptotephra analysis, taking samples and doing heavy liquid separation, but it can still help to recognize more tephras than you would typically see just by looking at a core. And it's in an automated way, which I think is always useful. So here's one that is interesting for this group, right, about Varves. There's a couple papers that have shown you can actually produce full grain size distributions from the hyperspectral data. In this study, they use partial least squares regression. They're using both the visible near infrared and the shortwave infrared sensors, and they take all of the spectral data, so not an index, but the full range of reflectance data, put it into a partial least squares regression model, and produce grain size distributions as their output. And in this study, they've identified a few different grain size in members that they could link to specific types of sediment. So here, this one is associated with diatoms and large calcite crystals, which are similar grain size, small calcite crystals, and detrital sediments. And using that in these varves, you can see there's like 5 varviers from Lake Bourget in this figure. They were able to produce maps of these layers of these different types of sediment, right? Detrital in yellow, the diatom and calcite, large calcite together in blue, and the fine calcite in red. So making a map of the seasonal pattern within the varves, I think, is a pretty powerful tool, basically like a microphases analysis, but in an automated way. They were able to even calculate accumulation rates of each of these 3 types per year. So this is pretty interesting for the types of work that we in the Varves Working Group are familiar with doing with a microscope. Now you can do using sort of just algorithms on data, right? In this study that I did on Lake Shabinsky, I don't want to talk too much about Lake Shabinsky because we've already heard about it before in this seminar series, right? But a little bit about Lake Shabinsky. In this study, we did a very detailed investigation of the last 60 years or so, where we had both the micro XRF imaging data from the Tornado Bruker and the hyperspectral imaging data. And you can see how powerful these data are to see very high resolution, sub-annual scale changes in the sediment. And what I was interested in doing is, can we use the sub-annual time series to basically classify varve types? So we were able to do that using a multivariate time series clustering algorithm, right? So looking at how all of these variables, calcium, phosphorus, total chlorophyll, bacteria, phaeophytin, iron, etc., how all of these variables change throughout a single varve year, right? We aligned all of the varves at the beginning of the calcite precipitation and put them on the same scale, and then said which years look similar to one another based on all these variables and their pattern across the year. And doing that, we found 4 different varve types, each with their own different characteristic. And what I found really interesting is that we could see that the varve types were associated with different seasonal weather conditions. So for instance, in particular, we saw that certain varve types, varve type 1 and varve type, was it 2, I think? 2 and varve types 1, 2, and 4 in particular were associated with warmer temperatures in the spring, and they were associated with more calcite precipitation in the spring and summer, I should say, right? So we saw significant changes in temperature and wind when we just group years based off of these varve types that came from the high resolution time series. So I know that's a fast overview, but just giving you quick examples of the kinds of things you can do with this data. Another thing that we've been really interested in our group in Bern is reconstructing lake productivity and water column oxygen over the long term, over the Holocene, trying to put in context the eutrophication that's happening in recent decades. So we went to a number of sites throughout Europe, and we measured chloropigments as a proxy of aquatic production, and this pigment called bacteria phaeophytin A, which is produced by purple sulfur bacteria. And it's a really useful indicator of anoxia because these bacteria live in sulfitic waters, but they are photosynthetic bacteria. So they live in sulfitic waters, anoxic waters that also receive light. So they're always living basically at the chemocline of a lake, and they only grow when the chemocline of the lake is shallow enough to be within the photic zone. So it tells you that the anoxic zone was extensive and relatively shallow, and that the lake was really strongly stratified when you have this pigment, bacteriophyophytin A. And here's an example from Lake Mose. Mose has a really great record of early human occupation of the site. There's archaeological findings, and there's a really nice pollen record as well, showing the human activities. And so we compared the pollen data with our pigment data at high resolution. And zooming in a little bit here in this lower part, you can see these periods of human occupation in the blue down here. And they're typically associated also with a spike in charcoal. They were clearing the forest and making fires. This brought probably a lot of organic matter into the lake. And then what happens is that the humans move away and the forest recovers quite quickly. So if you look here at the green line, you see that the tree pollen was less when humans were there, and then it recovers. And it's when the forest recovers that we actually see the blooms of the purple sulfur bacteria and the bacteriophyophytin A pigment. So the lake becomes anoxic, probably due to an input of organic matter, but also very important is the forest shielding the lake from wind mixing. This lake is relatively small in surface area and deep. So if there's tall trees around the side of the lake, it really prevents the lake from mixing because there's little wind activity on the surface. And this is repeated several times where you have a charcoal spike, you have trees, you have forest clearing, and then forest recovery. And during the forest recovery, you see that the lake is more anoxic. And this wasn't found just at Mose. We found this at a number of sites. So these are figures taken from a variety of sources. So I apologize, they might be a little bit difficult to read. Here's an example from Lakes Łaździuny and Szabinski in Poland. And in purple and black, you see the bacteriophyophytin records. Over here, you see the tree pollen records. And deforestation occurred much later at these sites. In northeastern Poland, there was very little human activity until 1600 AD, or the human activity was light on the forest. And then we see major deforestation at 1600 AD, and it's at the same time that the bacteriophyophytin decreases and goes away. Previous to that, the forests were closed and the lake was mostly anoxic. Then another example from Switzerland at Solpensee, it's a similar pattern. Deforestation happened around 200 AD. So it starts around here, and you see again, that's when the bacteriophyophytin disappears. The lake started to mix more because there was less trees surrounding the lake. At Lake Mezzano in Italy, we see similar patterns. It's a little bit more complicated because there's an early and variable impact of humans. Since around 3,800 years ago, you start to see these forest clearings, right? And typically when we see the forest clearing, there's a response in the bacteriophyophytin where it decreases, and the long-term trend is towards less trees and less bacteriophyophytin. Less trees means more mixing of the lake. So it seems to be a consistent response for these, in particular, small, deep lakes, that forest shelter is a really important control on the mixing of the water column and the oxygen in the water column. So that's an interesting result that we've been able to obtain using the hyperspectral imaging method and comparing with other proxies. Lake Szabinski, one more example, just to show this at detail. Looking now at the last 2,000 years in this plot and zooming in on this transition phase, we had a similar thing like in Lake Mose, where there was some human activity here, and they move out of the area, and you see a slight increase in the tree pollen, and that leads to these blooms in the purple sulfur bacteria. Here's how it looks in the core. This is about 100 years of Varves sediments in Lake Szabinski. Here I have the total chlorophyll, so lake productivity proxy. There's not a big change across this interval, but you see this really sudden onset of the purple sulfur bacteria blooms. And I just think this image is powerful because it shows how useful the hyperspectral imaging data is. You see exactly when this starts. You can see within single Varve years, there starts to be little blooms, and then there's really extensive blooms of the bacteria. And it's something that you wouldn't be able to recognize whatsoever just looking at the core phase. Right. Here's going to go in a little bit more detail on a new study that I've been working on from Lake Cadagno in the Swiss Alps. And actually, I just submitted a manuscript on this, so I'm kind of excited about it. The preprint posted literally like 2 hours ago to Biogeosciences. So if you're interested, you can go to this link or just search the Biogeosciences journal website and you'll find a preprint about this one. Lake Cadagno is an interesting site in the Swiss Alps. It's an alpine lake, and it has this interesting setup where most of the surface water inflow comes from a catchment that's dominated by gneiss and crystalline rocks. And so it's very low in solutes, very clear fresh water. And then it has a significant groundwater source from the other direction, which passes through this dolomite, grey-wacky Peoria zone of the Alps. And the groundwater is extremely rich in solutes after passing through this bedrock, and it's particularly rich in sulfates. And that leads to a very dense monomolybdenum. Bottom waters are very dense, and they're not mixing with the upper epilimnion, where it's fresh water and it's much less dense, right? So it creates this setup where you have a very strong chemocline. Here's profiles of the water column, and you have a lot of hydrogen sulfur in the lower layer, right? It's very sulfidic. And this site is well known for this, and it has these really dense bacterial blooms of the purple sulfur bacteria and the green sulfur bacteria. So these are both two different kinds of these phototrophic anoxygenic bacteria. They both need sulfur, sulfides as part of their photosynthetic process, and they need light. The purple sulfur bacteria in particular need light. The green sulfur bacteria can live in pretty dark conditions. They've been found, I forget exactly what depth, but maybe like up to 100 meters depth in the Black Sea, I think, really deep. So they can live at pretty low light conditions. So in Lake Cadagno, you typically have the purple sulfur bacteria on top and the green sulfur bacteria in a layer below, because the purple sulfur bacteria need more light, right? So we were interested in going there to test the hyperspectral method on a lake that has both of these types of the phototrophic bacteria, because we hadn't yet really found that we could measure pigments from the green sulfur bacteria. And measuring, so the green sulfur bacteria, they produce bacteria chlorophyll C, D, or E, depending on the exact species of the bacteria. And these types of pigments are difficult to measure with other simple, rapid techniques, because if you extract them in solvent, they look extremely similar to chlorophyll A. Their spectral profile when they're dissolved in a solvent looks almost identical to chlorophyll A, making it really hard to measure these. But when they are in sediments in situ, when they're not dissolved, they have a distinct spectral profile. And what we see in Lake Cadagno is an absorption trough at 715 nanometers, which is in agreement with what we'd expect for bacteria chlorophyll E. So here's showing some examples of different samples I took from the Lake Cadagno core. This sample shows an absorption trough associated with chlorophyll A. This sample shows absorption associated with chlorophyll A and bacteria phaeophytin A, so the purple sulfur bacteria. And this one shows chlorophyll A, bacteria chlorophyll E from the green sulfur bacteria, and bacteria phaeophytin A. And you can see, right, so these are the spectral profiles. I'm using continuum removed reflectance, so it looks a little bit different. It just highlights more of these absorption troughs. And you can see how it compares with the indices here, where this is this new index RABD 710 to 730 max. which should be associated with the pigments coming from green sulfur bacteria. So we compared with measurements done on the HPLC, and our calibrations are working as we expect for bacteria phaeophytin A and total chlorophyll A. It was difficult to quantify the bacteria chlorophylls in the HPLC data. So I compared with a different pigment called isorenarotene. This pigment is produced by all the different species of green sulfur bacteria that are living in Lake Cadagno. So it is a good proxy for the green sulfur bacteria. And we see a strong correlation between the RABD 710, 730 and the isorenarotene, which says that our RABD method should work as a proxy for the green sulfur bacteria, since the green sulfur bacteria are all making this pigment and nothing else is making this pigment. So here's what our record looks like, going into a little bit more detail from the record from Lake Cadagno because it's new and exciting. Time is to the left. In green here, it's RABD 670, which is chlorophyll A, and it's tracking productivity in the epilimnium, basically. Then we have our green sulfur bacteria proxy and our purple sulfur bacteria proxy, also comparing these to flood data and molybdenum data from previous studies of Lake Cadagno, as well as some climate records. And just briefly to go through some of the things we see that there was a continuous presence of the phototrophic sulfur bacteria, either or both the purple and the green, since about 9,800 years ago, which confirms earlier results based on the molybdenum record and low resolution pigment data. It confirms that the lake was truly meromictic and sulfidic throughout the Holocene. But we do see interesting changes. We see that the purple sulfur bacteria are particularly dominant in this time period from 8.8 to 3.4 thousand years ago. And then there's this shift at about 3.4 Ka, where the purple sulfur bacteria become much less abundant and the green sulfur bacteria become more abundant. And we interpret this change, it could be due to a number of factors. It's probably related to the fact that people moved into the area and started clearing some forests. That increased also the flood input. We also have a climate shift here, cooler temperatures associated with the neoglacial. All of these would together lead to conditions where the lake is more prone to mixing and potentially the chemocline could become lower. Additionally, we think that there was probably less solute in the groundwater. We think it was a little bit wetter time and greater through flow of groundwater through the bedrock, faster through flow that is, that leads to more dilute groundwater. So you have probably a less strong gradient between the bottom and the top layers of the lake. And what this would do again is probably lower the chemocline and have less light available at the chemocline if it's at a greater depth. And that is not favorable for the purple sulfur bacteria and more favorable for the green sulfur bacteria. When there's plenty of light available at the chemocline, the purple sulfur bacteria dominate and they shade out the green sulfur bacteria. The green sulfur bacteria can still grow, but at much lower abundance when they're shaded by this dense layer of the purple sulfur bacteria. And when we look at really at detail at some of these flood layers, and there's also mass movements in this core, we see that the green sulfur bacteria tend to recover much more quickly than the purple sulfur bacteria. And again, this is probably due to light availability at the chemocline. If there's a flood or a mass movement, you might have more turbid waters, less light, or you have the chemocline be disturbed and lowered, which also leads to less light. Okay, so that was a lot, I think. To summarize in a simple way, hyperspectral imaging is a cost-effective, non-destructive, and rapid technique to obtain ultra-high resolution biogeochemical data from lake sediment cores. It's suitable for high-frequency variability or short-term events, but it's also suitable for long records and core logging. You can obtain a wide variety of sedimentological and chemical information, and calibration models are increasingly being used to produce semi-quantitative data. And these calibration models and new proxies with this method are still being developed. It's a relatively young method, so we're still learning what we can gain from this method. I want to share some acknowledgements because this work depended on a lot of people. So first, my PhD supervisor, Martin Grosjean, who had, I think, really the original idea to use hyperspectral imaging on lake sediment cores. I also want to point out Christoph Boots, who developed our system in Bern, including the software that we're using to analyze the data, and that is absolutely essential for everything that I've done with the hyperspectral imaging system. So thank you for your attention, and I'm happy to take any questions.